So let's get into the actual biological molecules that I've been mentioning for the last 40 minutes. So the major classes of biological molecules are all molecules that are already familiar to you, either because they're a part of our diet or they've been in the news or whatever. They're all familiar to you. So we've got carbohydrates. These are broken up into sort of two big groups, monosaccharides and polysaccharides. So mono meaning single or one, so one saccharide or one sugar, and then polysaccharides, which are multiple sugar molecules linked together. Lipids, and while you may not be super familiar with that term, lipids are a group of molecules that consists of fats, which I'm sure you're familiar with, phospholipids, and steroids, which you are probably also familiar with, at least familiar with the name. Then we have proteins and nucleic acids, which are DNA and RNA. So as I said earlier, there's a lot of information in this chapter. I would strongly encourage you to make a summary chart of some kind. We can work on this together in class if you want, but definitely do some flashcards or make a chart, something to help you keep these all straight. So let's start with carbohydrates. So carbohydrates are sugars or polymers of sugars. So you probably all know about eating carbs. Carbs are always the best one. They're delicious bread and they're pasta and they're sugar and they're sweeteners. Carbohydrates make up a huge part of our diet, good or bad. So they come from things that are plant-based. So things like flour. We've got beans, oats, sugar itself, like glucose or fructose. All of these are plant-based and they're just delicious. Another big one is corn. So I've got a picture down in the bottom that's got a lot of pasta, beans. There's some tortilla chips, a lot of bread, things like that. So as I said when we were talking about the structure of carbon, one of the things that's important about carbon is that the bonds that it forms with other carbon molecules are really high energy. And so it makes sense then that one of our most important biological molecules for energy is going to be carbohydrates that are basically long chains of carbon. So we have two types of carbohydrates. We've got our monosaccharides. These are our simple sugars or single sugars, and they are going to be the building blocks for our more complex sugars. Now, if you're looking at the chemical formula of a sugar, the way that you're going to identify it is by this basic formula. Sugar formulas are always going to be multiples of CH2O. So 1 carbon, 2 hydrogen, and 1 oxygen. Multiples of that. So for example, glucose, you've probably heard of glucose before. That is a single basic, very common sugar. It's the one that we find most often in plants. When plants are going through photosynthesis, they're building something called G3P. That G3P is going to be very quickly converted into glucose. So glucose, we can recognize our formula in there, CH2O, a multiple of that. So we've got 6 carbons, 12 hydrogens, and 2 oxygens. So the general structure of a glucose molecule is going to be a ring. And that ring is going to have at least 2 oxygens coming off of it. Don't pay attention to my exact structure here. I'm just randomly placing oxygens. But glucose is going to have that basic CHO formula. So monosaccharides can be found in a linear or a ring structure. Usually the ring structure is a little bit more stable, and so they won't stay in their linear structure very long, but it's easier to look at a ring structure, a linear structure, to see what's going on, to see what's bound there sometimes than it is to look at the ring structure. But we've got a couple examples of our monosaccharides here. You'll notice something about them. You've got your carbons as a chain in all of them. You've got your hydrogens everywhere there's not something else bound to a carbon, and then you've got lots and lots of oxygens. So those hydroxyl groups and those carbonyl groups are really important because those groups are going to interact well with water. This is why sugar dissolves well in water, because we've got a lot of oxygens. Those hydrogens can be kicked off, and then we'll get a negative charge there. We get partial negatives because of the electronegativity of oxygen. So these guys react really well with water. You'll look down here, or you'll notice down here, glucose and fructose. Those are two common monosaccharides. They're very, very similar. The major difference is the placement of this functional group. So there we go again with our structure and our function. We've got a slightly different structure, so we get a slightly different sugar. The placement of those functional groups is going to affect how we make our ring structure. Now, you do not have to understand how we make the ring structure. We're going to leave that for our chemistry teachers. But the important thing here is that these linear forms will basically fold in on themselves and produce a ring structure that is much more stable than that linear structure. And our carbohydrates, our big polysaccharides, are going to be made up of these rings. You know, the important thing that you need to know about these rings is that we've got the carbonyl groups and the hydroxyl groups. I'm sorry. The carbonyl group is not part of what I'm saying. The hydroxyl groups are off of the outside. Those hydroxyl groups are what are going to be involved in bonds between the different rings. So those hydroxyl groups are going to be important for building our polysaccharides. So our monosaccharides, are specifically glucose, are the major energy storage molecule for the purposes of maintaining and storage in our cell. Glucose specifically is one of the major nutrients in our cell. It is broken down in cellular respiration. And when we break down that glucose, we release energy. And we actually can use the energy in that glucose to produce ATP. And that ATP is going to be what is powering all the processes of our cell. If you're paying attention, you will notice that I said we're making ATP to power our cells. Just as a little side note, this is all occurring in the mitochondria, because the mitochondria is what? The powerhouse of the cell. The process of photosynthesis, which is in a very loose way the opposite of cellular respiration, is where we build glucose to store energy. So our plants do this, and then we eat the plants, and then we get the energy from the plants. So plants are building up glucose, this really important monosaccharide for us. One step up from a monosaccharide, so one step up from one is two. A disaccharide is one step up. A disaccharide is two monosaccharides that are joined together. In the last video, I said that they were monomers can be, joined together by a dehydration reaction. Remember that a dehydration reaction is a reaction that is going to release water to form a bond between monomers. So we can build disaccharides or even larger molecules, polysaccharides, by a series of dehydration reactions. So down here on the bottom, we've got a glucose monomer and a fructose monomer. We put those together with a dehydration reaction. So we make a water molecule and we release it. When that happens, we form a bond between our two sugars. That is called, in this case, a glycosidic linkage. It is the bond type that binds together our saccharide molecules. So when we bind together a glucose and a fructose, we get sucrose. You've probably heard of that one before. We can continue to link those molecules into really, really long chains. Two examples of really, really long chains are starch. Find starch in potatoes and corn, things like that, and cellulose. Cellulose is the structural molecule in plants. It is what forms the rigid structure in our plant molecules. It allows them to stand up without something like bones like we have. So for each of these molecules, I am going to talk about the bond type. I would like you to know the name of the bonds. And they're all built in the same way. They're all dehydration reactions. They just have specific names based on what the molecular level is actually happening, what molecules are being bound together. I would just like you to know the name, and I would like you to know what's happening. I would like you to understand what a dehydration reaction is. So the structure and function, again, are determined by the monomers and the position of those glycosidic bonds. So sucrose is very common in our diet. It's very easy to break down. There's just one bond we have to break to be able to use it to break it back down into glucose and fructose. And then of course we can use glucose to help us produce, or to help us produce ATP. Bonds in glucose are going to be broken and we can get our energy, our cellular energy, from those bonds that were in the original sugar molecule. But it's not just the monomers, it's also the position or the shape of that bond that forms that affects the structure of our carbohydrates. So there are a couple of major functions for our carbohydrates, but those two big ones are going to be structural function and storage function. So this really complicated busy figure is just showing you a couple of different ways that the monomers of glucose are linked together. And if you look really closely, you'll see some very subtle differences. So up on the top, we have starch. We've got potatoes as our example for where we get starch. But starch is a relatively simple molecule. It is just a linkage, continuous linkage, straight line linkage of glucose monomers. So if you look really closely at this picture, I know it's a little confusing, it's just a chain of glucose molecules. So it's just a long chain. And because it's a simple long chain, it's easy to break off the glucose, and that's going to allow us to get quick energy from starch. You'll notice that it's showing This second example that has a little bit of branching going on, that's just a slightly different, more complex molecule that's similar to the starch, still used for energy storage, but you'll see that the bonds are the same there. Down below, we've got glycogen. Glycogen is another energy storage molecule. This one comes from our muscle tissues. Glycogen is similar in that we've got chains of glucose with our glycosidic bond, but it's a little bit more branched. So you can see we've got branches coming off of the main chain, but we've still got chains of glucose that are going to be used. You can break them off. We can use them for quick energy stores in our muscles because our muscles need a lot of energy to keep them going. They're very energy intensive tissues. So these top two here, starch and glycogen, are our storage molecules. And then down below, we've got our structural. So our structural molecule is cellulose. I mentioned that just a minute ago. And the structure of cellulose, while very similar to starch and glycogen, has its own specific structure and therefore its own specific function. So cellulose forms fibers, and those fibers are really strong. Now, our bodies actually have trouble digesting cellulose because of the bonds that make up this molecule. We can't break them down really well. That's why fiber that comes from cellulose is so important. We actually need stuff in our body to help kind of flush out our systems, and the fact that we can't fully break down fiber helps to do that. That's why we, for digestive health, need a lot of fiber, because our body can't digest it, and it basically works as kind of like, I like to say, like a scrub brush. We can't break it down, and it sort of cleans out our system. But there are other organisms that can break down the cellulose, and so those guys are really important too. But because of the bonds, there's a lot of different interactions happening and a lot of bonds forming within the cellulose structure that give it its strength. And so cell walls of plant cells are actually made up of cellulose, and that gives plants their ability to stand upright because these cellulose fibers within the plant cells are strong and they're giving plant some structure. So you'll see here we've got sort of alternating glycosidic bonds here. That makes the function slightly different. And because of those alternating bonds, we actually have some hydrogen bonds that can form in between the strands of cellulose. So not only do you have covalent bonds linking all of your sugar molecules, but you've also got hydrogen bonds in between those strands of cellulose molecules, and that's going to give it some extra strength. Remember in chapter 2 we talked about hydrogen bonds and how individually they're really weak but collectively they're really strong. That's one of our special properties, or we get one of our properties of water because of hydrogen bonds. Those water molecules are attracted to one another because of hydrogen bonds. That makes them collectively strong, so we get things like surface tension. The same idea is happening in these cellulose molecules. We've got hydrogen bonds occurring between the strands of our cellulose, which helps to make those fibers even more strong, which is why we can use them for structures. So we've got our two major functions, structural and storage polysaccharides. We've got our starch and our glycogen for storage, and our cellulose for structural. There's another type of structural molecule. This is chitin. So chitin is, you'll notice, a little bit different. We've got this functional group up here with an amine group on it. But this chitin is also important in structure. This is the basis for the shells on many of our arthropods and things like that, many of our insects and crabs and things. And if you've ever touched any of these insects, you'll know that they're really, or in any of these insects or animals, you'll know that these structures are really hard, and that comes from this linkage similar to our cellulose of these carbon ring structures. So next we have our lipids. So lipids are a unique group within our biologically important molecules that have slightly different properties than the other three. And there are a couple of types of lipids. They are fats, phospholipids, and steroids. So lipids are a little bit different because they're not true polymers. Our other three molecules are all monomers linked together in really long chains. Different molecules, different chains, but they're all long chains of molecules. Our lipids are not polymers. They are smaller molecules, but the reason that we include them in our biologically important molecules is that they're biologically important. Even though they're not polymers, they have really specific functions that are really important in our cells. So there are several different kinds of lipids. They make up a diverse and very specific group with some specific characteristics. They tend to be much smaller than our other long chain polymers, but they are grouped together, even though they're all a little bit different, because they are all hydrophobic, which means they don't play well with water, right? They are water fearing. They don't want to mix with water. And you'll recognize this when you think about what some of these lipids are. So the ones that you know of are fats. So common fats in our diet are olive oil or vegetable oil. I'm not sure what that one is, but olive oil, vegetable oil, canola oil, things like that. There are fats in some of our meats and what we would consider proteins. But there's also a lot of fat in our meats. There's fats in things like butter, avocados, beans, nuts, things like that. I'm not entirely sure why there's broccoli on here because there's basically no fat in broccoli, but maybe they thought it looked nice. So you are probably all familiar with fats, and you're also probably familiar with the idea that fats don't mix well with water. So if you've ever tried to wash a pan of bacon grease, you know that it doesn't mix well with water. Or if you've ever poured oil into pasta water, you know that it floats on top. You are probably not as familiar with phospholipids. We're going to talk about those guys. And then steroids, of course, you are probably familiar with them because you're familiar with the idea of steroids and hormones, but they are their own unique type of lipid. So the lipid that you are most familiar with, and the one that we're going to kind of talk about the most today, are fats. So again, fats come in our diet. They usually, but not all the time, come from animal sources because fat is one of the major ways that animals store their energy, but we can absolutely get them from plants as well. So vegetable oil, canola oil both come from plants. Olive oil comes from plants as well. So on the bottom here, we have a fat molecule, also called a triacylglycerol. That molecule there is one single molecule. That's the whole thing. Very different than our carbohydrate, and as you'll see in a few minutes, very different from our proteins. But fats all have a similar structure, which is why we include them in our discussion of these polymers, large molecules. Fats are 3 fatty acids. So on the top here we have glycerol, which is one of our fatty acids. You'll see that on the left side, we've got triglycerol. Let me back up. We have a fatty acid that is going to be combined to make a triglycerol. Each unit of the triglycerol has a glycerol molecule. So here's our carbon base chain again, attached to a fatty acid. So a really long carbon chain. It's called an acid because of this functional group over here. That's our carboxyl group. Carb, oxygen group, our carboxyl group. The diagram here shows that this is being formed by a dehydration reaction between the glycerol and the fatty acid chain. Now, you know that the major function of fat is energy storage. So if you bring in too many calories, too much energy into your diet, your body is going to store it as fat. The reason that your body stores energy as fat is that it's really compact storage. And the reason that animals in particular store a lot of fat is that we have to move around. And so unlike plants who can use carbohydrate as a lot of their storage that make these big, huge molecules, we need, and other animals need, a really compact way to store our energy. So fats have these really high energy hydrocarbon chains at the end. Remember at the very beginning of the chapter, I talked about hydrocarbon chains and how they have tons and tons of energy. Well, here we are seeing that in our fat molecule. So we've got this glycerol backbone. We're going to add 3 fatty acids to make our fat molecule, this triacylglycerol. So this is sort of just a basic fat molecule. In order to make this fat molecule, we have 3 dehydration reactions, one that occurs at each oxygen on that glycerol to link in the fatty acid tail. That linkage or that bond is called an ester linkage. So you've got 3 of those to make one single fat, but then that's it. This is your fat molecule here. So fat molecules come in all shapes and sizes. There are 2 major groups of fats. One is saturated fat, and you have probably heard about saturated fats in your diet. Saturated fats are the quote-unquote bad fats. You can identify a saturated fat because it is usually solid at room temperature. So think butter, bacon grease, lard, things like that. When you cool them down to room temperature, they become solid. You can melt them very easily, but they become solid at room temperature. So the reason that they are bad for you is that these molecules can pack in really tightly into your arteries and things like that. And so you want to have a diet that is low in saturated fats. The reason that they pack together so tightly is we have this really nice regular chain of hydrocarbons all in a neat little row. They lay down together really nicely, so they stack really nicely together. The reason that they are long, beautiful hydrocarbon chains is that there are no carbon-carbon double bonds. So each one of these points is a carbon, there's a single bond in between all of them. On the other hand, an unsaturated fat means that you don't have all of the hydrogens there that you could have because you have at least one carbon-carbon double bond. Remember at the beginning of the chapter, I said when you have a carbon-carbon double bond, the molecule moves from being a tetrahedron shape to a flat shape. That's what's happening here. That flat shape around that double bond is causing a little kink, and that keeps it from packing so tightly. So your unsaturated fats are the fats that are going to be liquid at room temp. So olive oil, vegetable oil, sunflower oil, all of those oils that you might find in your pantry in liquid form are going to be your unsaturated fats. Now, they tend to be a little bit more healthy because of that kink. They can't pack so tightly in your arteries and things like that. But there's a little caveat to that, and that is the distinction between cis and trans fats. Remember we talked about isomers a little bit ago, where we have our carbon-carbon double bond. Cis molecules, cis molecules have our functional groups on the same side, whereas trans molecules have our functional groups on the opposite sides. This is a really important distinction in our diet. So if you look at this molecule here, we've got a cis double bond. You can see the functional group coming off the double bond gives that molecule a particular shape. We get this little kink. Trans fats are basically saturated fats who have been chemically processed to put in a double bond. So you get the kink kind of the other direction. And you might think, well, that doesn't sound like a bad thing. It's not going to pack in as tightly. The problem is that because trans fats are artificial, they're artificially produced, our body doesn't know how to deal with them. And so they can be really unhealthy if you have a diet high in trans fats. And trans fats tend to be in our really unhealthy, like processed foods, which among other reasons is why they are so bad for you, is because they may be high in trans fats and our bodies don't know how to deal with those. So next we have our phospholipids. Phospholipids are really important for our cells. We're going to have basically a whole chapter on our phospholipids, chapter 5. So I'm not going to spend a ton of time on them now, but phospholipids are important because they make up the cell membranes of all of our cells. They're slightly different from our fatty acids, although the idea is the same. They have a glycerol molecule. They have two fatty acid tails coming off. There's a little cis bond kink in it. Instead of having the third fatty acid, though, they have a phosphate group. Now, having that phosphate group gives this molecule some really important properties. One, this charged area with all those oxygens up there is going to be polar and it's going to be hydrophilic, which means it's going to interact with water. Down here on the bottom, because we have those fatty acid tails, they're going to not want to interact with water. They are going to be repelled by water. So this is a really cool property of these guys, because when you put them in water, the heads are going to interact with the water. The tails are going to turn inside and face one another. So you get these layers that are hydrophilic on the outside and the inner layer that is hydrophobic. Now, this is important because it's going to create a barrier for the outside of our cells. It's basically going to allow us to have cells that separate internal and external environments. So we're going to come back to these and talk about them in a couple of chapters. All of that made me realize that I forgot something. Fats are hydrophobic. That means they don't interact well with water. Now, the reason for that are these long chains of hydrocarbons. Remember that those chains of hydrocarbons are non-polar covalent bonds. So they are going to generally be completely hydrophobic. If you mix oil and water, you all know this, you mix it, you try to shake it up, you think of like Italian salad dressing, things like that, you're going to get a layer of oil and water, and that's because the oil is being repelled by the water. Our last group of lipids are the steroids. I'm truly not going to talk about these too much because we don't really use them again in Biology 101. if you continue in Biology 102, you'll actually talk about hormones and things. And so the structure will be important there. But steroids are made up of 4 fused rings. Again, those are carbon-rich rings. And steroids are distinguished by their chemical groups. So their function is determined by what is attached to them. Cholesterol is the basis of our hormones. So we usually think of cholesterol in a negative context, but we actually need it in our body for a variety of different reasons. One of which is that it is used to produce our hormones. So this molecule on the left is a cholesterol molecule. You can see that the basis for that molecule is really similar to the products that come out of it, which are, in this example, are estradiol and testosterone. Talked about these guys when we were talking about chemical groups. So again, you can see that the backbone is the same, the structure is the same as the cholesterol, they're the same as each other, but have slightly modified functional groups. So in order to make estradiol and testosterone, these functional groups are modified for their specific function in the body, which estradiol and testosterone are, of course, our sex hormones that help our bodies to produce male or female development and characteristics.